So I think we have to start.
Yes, even I do think that we have to start. Okay, fine. So let's just start with that and let the other to join us. So try to understand first what is the oppressed part, what is the non-oppressed part, because that is a more important. In previous class, I was trying to understand, but so look, we'll again discuss the chloroplast in that. Again, we'll discuss again the chloroplast, and when I'll teach you the that cell biology part, the structural unit of and like this kind of the chapter. So basically, that is a cell biology. So cell biology is made up of three parts. That is a cell organelle, cell and cell organelle, and the second thing biomolecule, and third thing is cell cycle. So the cell organelle part there, I'll again discuss this part in detail. But here, just I'll give you the glimpse of the things which is related with the photosynthesis. So basically, this is the structure of the chloroplast, and out of this, in this structure, outer membrane, inner membrane, and there is a space between the membrane that is called intermembrane space. Right. In this intermembrane space, one more thing which I want to add on here: electron that the uh, electron transport chain will go on right in this surface inner membrane, and what will happen? The formation of ATP will take place. Right. We'll discuss later. Leave it. So look, those part there are some connections between the two. Before that, yes, this pile of the this structure or this pile up is called granum. So granum is a singular word. So the all the granum collectively known as the grana is the plural word. So if you'll ask me, then 1, 2, 3, 4 and 5, so there are total 5 grana, right? And if you ask this single structure or say this single pile, that I'll call granum, right? Granum is singular word. There are some connections which connect one thylakoid, one granum to another granum, right? These structures are called stromal lamellae. Clear? Now try to understand what is the oppressed part and what is the non-oppressed part. So basically, those part which are or those surfaces which are, so this single structure, this single coin is called thylakoid, right? So number of the thylakoid when pile up one upon another, they make that structure, that a group of thylakoid, that is called grana, right? So the part of thylakoid which is in contact with another thylakoid, that part is called oppressed part, right? So that part of thylakoid which is in contact with another thylakoid, that is called oppressed part. This oppressed part contain photosystem segment. What is the photosystem? Photosystem is basically a group of pigment molecule, or rather than you can say group of chlorophyll molecule is called photosystem. So in this is called oppressed part, right? In these oppressed part, what is the oppressed part? Oppressed part of that part of thylakoid which is in contact with the surface of another thylakoid. That is called oppressed part. What is the non-oppressed part? Those part of the thylakoid which are directly in contact with the stroma, means they are the open part. They are called non-oppressed part, like this one, right? Non-oppressed. Non-oppressed part contain photosystem one, right? Photosystem. And uh, again, I have tried to show this concept in the another diagram. In another diagram, you can see this part of the thylakoid. This is thylakoid membrane. This is structure is thylakoid membrane. The space inside the thylakoid is called thylakoid lumen, and these part which are in contact with another thylakoid. So these part called appressed part of thylakoid. So appressed part are those part which are in contact with this, which are in contact with this. This is called appressed part, and those part which are open, they are called non-appressed part. And I have already told you that the oppressed part will always have photosystem second, right? And non-appress part of always have photosystem first. Photosystem second is also known as P680. Photosystem first is also known as P700, right? This P60 photosystem second or P680 is responsible for photolysis. Photolysis of H2O. Photolysis of H2O is also known as the photo means light lysis by breakdown, right? That is breakdown of breakdown of H2O molecule, right? And this P700 it do not involve the breakdown of water. Is that clear or not? Tell me.
Yes, sir.
Okay, that thing you need to remember. Right? Uh, I'll dictate something in next PPT. First, let me make you understand, fully understand, right? Then I'll dictate. Look,
So,
yes, please tell me.
so what is meant by P680
What is
and P700?
P700, P680? I'll tell you. I'll explain it in more detail. Just here I am going to. I just want to tell you the appress and non-appress part in previous slide. That's what I want to convey with you, right? I'll tell you what is the P680, what is the P700. Basically, they are a special kind of the chlorophyll A molecule, right? I'll explain that thing. Now you'll get more clarity. Look, now this is a single thylakoid. This is the lumen of thylakoid. Look, these are the oppressed part, right? They are the called appressed part. These appressed part, they contain photosystem second. Along with photosystem second, there is a red color point, this red color structure. This red color structure is always found with the photosystem second. You will always see this red color structure. This red color structure, which is shown over here, this is called oxygen evolving complex, OEC. This is responsible for photolysis or breakdown of the water molecule, right? This is oxygen evolving complex. It is responsible for photolysis. So photolysis takes place, water molecule breakdown, formation of oxygen molecule, two hydrogen atom, so that is 2H2, two 4 hydrogen atom, and 4 electrons evolve over here, right? That's how the photolysis of water takes place, right? It takes place in only in oppressed part. In non-oppressed part, photosystem 1 is non-oppressed part, means open part. Non-oppressed part also have ATP synthase. ATP synthase is enzyme which is responsible for formation of ATP, adenosine triphosphate, from adenosine diphosphate. Right? ATP synthase enzyme responsible for formation of ATP. The process of formation of ATP is also known as photophosphorylation. Photophosphorylation is the process of formation of ATP that take part in non-appressed. Is that clear?
Yes, sir.
Sir, can you please repeat?
Yes, sure. These are the appress part. What are the appress part? Those part which are in contact with another thylakoid, right? They always have this. So photosystem second. Photosystem second is always found in the where appress part, right? Along with the photosystem second, you will always see that oxygen evolving complex, OEC, right? This oxygen evolving complex, they break down the uh, water molecule, right? They break down the water molecule, right? Due to breakdown of the water molecule, uh, oxygen comes, hydrogen comes, and 4 electron come. The process of breakdown of the water molecule is called photolysis. Is that clear?
Yes, sir.
First, please write it down the same. Note down. Photosystem second, photosystem second or P680 are always found at appressed part of thylakoid. Full stop. PS second means photosystem second. PS second always contain oxygen evolving complex OEC. PS second always contain oxygen evolving complex in bracket OEC. Full stop. OEC help in OEC help in breakdown of OEC help in breakdown of water molecule, that is photolysis of water, which takes place in appressed part only. Full stop. So I think this point is clear to you, right? Clear to both of you?
Yes, sir.
Yes, sir.
Great. Now come to this part, non-appressed part. So non-appressed part is it remains in the contact with the stroma, means gel, right? Because oppressed part remains in the contact, like this was one thylakoid, this is another thylakoid. So both are in the contact. So this surface, which is in contact of this surface, this is called oppressed part. Oppressed part is not in contact with stroma, right? But these external part which are in contact with the stroma, they are called non-appressed part. So non-appressed part, they always contain ATP synthase enzyme, right, which help in formation of ATP from ADP, and this reaction is called photophosphorylation. And photosystem first is also found in non-appressed part, right? So please note down. Non-appressed part contain non-appressed part contain ATP synthase enzyme, non-appressed part contain ATP synthase enzyme, which is responsible for formation of ATP, which is responsible for formation of ATP from ADP, which is responsible for formation of ATP from ADP. Full stop. This reaction is called photophosphorylation. This reaction is called photophosphorylation. Full stop. Photosystem 1, oblique P700 is also found in non-appressed part. Now, is that clear? Both of you, Aisha and Rabia?
Yes, sir.
Great. Now I will tell you what are the photo system. I told you that a photo system. Now you will come to know what is the P680, P700. Right? Try to understand. Both of the photo system, they have the different composition, but their structure is almost same. This is the diagram of photo system. In the photo systems, first and second both look like in like the same diagram. They represent both of the photo system. Now try to understand. Photo system 1 and second both are the combination of 250 to 400 pigment molecule, right? In pigment molecule in center there is a reaction center that is called chlorophyll A molecule, and there is a primary electron acceptor which accepts the electron. The combination of both reaction center and primary electron acceptor both are collectively known as the reaction center, right? Uh, Try to understand. This reaction center is made up of two chlorophyll A molecules. That is a dimer of chlorophyll A molecule. This chlorophyll A molecule, this chlorophyll A molecule in photosystem 1 is known as P700, and because it received the wavelength of P700, 700 nanometer, and in photosystem second it is called P680. This chlorophyll A, right? That's why we call photosystem 1 as P700 and photosystem 2 as P680. Is it that clear?
Yes, sir.
Okay. Now try to understand what is this photosystem, how does it work? Look, this part is called reaction center. It is made up of chlorophyll A, dimer of chlorophyll A molecule and primary electron acceptor. Other molecules which are found around these reaction center, they are called antenna pigment. These antenna pigment are basically made up of carotenoid molecule, right? Carotenoid molecule. These small antenna pigment, they capture the photon, means they capture the energy, and they transfer energy to the finally to the reaction center. This they capture the small amount of energy, and finally they give energy to the reaction center. Reaction center collect a large amount of the energy, and it break down the water molecule here in PS2. Break down the water molecule, then electron is accepted by electron, this electron acceptor. That's how they work. Is that clear?
Yes, sir.
So the question was in the photos. Question was in the medical examination. What is the number of the pigment molecule that make a photosystem? And answer was 400 because 250 was 250 or 350 was not there. Right. So that kind of the question you also get. So I'm telling you whether that you need to know that what kind of the questions are being asked. Right. I'll give you some example of the previous year question. Right. Glimpse of some question. Look, photosystem 1 is found in photosystem 1 is found in non-appressed part. They have more chlorophyll A molecule. They have more chlorophyll A molecule and less chlorophyll B molecule. And carotenoid are found in the lesser. They are made up of a dimer of chlorophyll A molecule that make a reaction center, right? And this reaction center is P7. They involve both cyclic and non-cyclic photophosphorylation, right? Means electron transport. Photosystem second, PS second. Right. This PS second, it is located in non-appressed part of thylakoid, right? Non-appressed part of thylakoid. They contain chlorophyll A, chlorophyll B, and carotenoid. What is the difference between the PS1 and PS2? PS1 have more chlorophyll A molecule, less chlorophyll B molecule, but here the chlorophyll A and chlorophyll B are found in equal amount. Photo center contain oxygen evolving complex OEC. OEC is not found in the photosystem 1. So photocenter contain oxygen evolving complex and electron carrier. The next question which I've been asked in, twice or thrice, that is the composition of oxygen evolving complex. Sometimes the question I've been asked that which of the following is not the part of oxygen evolving complex. So oxygen evolving complex is made up of manganese ion, calcium ion, and chloride ion. So these three ions they make the oxygen evolving, and they are responsible for breakdown of water molecule, which is called photolysis of water. Is that clear? PS1 and PS2 clear or not? Tell me.
Yes, sir.
Now, would you tell me what is the anoxygenic photosynthesis? What is the anoxygenic photosynthesis? Tell me.
A
Aisha?
process, a
Yes, Sabir.
process where the uh, oxygen is not involved in the
Right,
reaction.
very good. Where oxygen does not evolve, right? So, by the way, I have written over here. So, where does it take place? Where does it take place? Tell me. Tell me some examples. Does it take place in plant or not? It does not take place in plant, right? Exceptionally, it takes place. So, anoxygenic photosynthesis is that photosynthesis where evolution of oxygen does not take place. If you want to note it down, you can. Right. So, anoxygenic photosynthesis. In anoxygenic photosynthetic, photosystem 1 does not involve, right? In anoxygenic photosynthesis, only PS second involve, right? Here, it takes place, for example, in bacteria like purple sulfur bacteria. So, purple sulfur bacteria they perform the anoxygenic photosynthesis where oxygen is not involved. Then, what does it involve? Then, evolution of sulfur takes place, right? The reaction is written over there. Here, the hydrogen donor is H2S, not the water, right? Is that clear?
Yes, sir.
Yes, sir.
Electron, please note it down. That is the question. Electron donor is hydrogen sulfide, H2S. No, please note it down. Anoxygenic photosynthesis, anoxygenic photosynthesis takes place in bacteria, like takes place in sulfur bacteria, sulfur bacteria, takes place in sulfur bacteria, full stop. Only PS second, only PS second is involved. Here, the electron donor is here, the electron donor is H2S molecule. Here, the electron donor is, that is a question. If you want, you can underline. Here, the electron donor is H2S molecule, not water. Full stop. Next point. Water is H2O. Water is electron donor. Water is electron donor in water is electron donor in oxygenic photosynthesis. Oxygenic photosynthesis. Okay. Now come to the next concept. Again here, chlorophyll, that is a photosystem are the group of the chlorophyll molecule. PS. PS1 is P700, PS2 is P650. In PS2, photolysis takes place. Now try to understand. Here, in PS1, the chlorophyll A molecule, it capture the 700 nanometer wavelength. That's why it is called PS P700. and electron acceptor is found, reaction center, right? And the rest, these blue color structure antenna molecule. Here in the PS60, the chlorophyll A molecule captures 680 nanometer wavelength. That's why chlorophyll PS second are also known as the P680. And there is a reaction center, and this is the structure. So tell me, is that crystal clear for both of you, Aisha and Rabia?
Yes, sir.
Yes, sir.
Biochemistry of photosynthesis. So photosynthesis is the combination of two reactions, light reaction and dark reaction, that we have studied, right? Light reaction is also known as the light dependent reaction. One more important thing I want to add on over here. Light reaction have one more name. What do we call it? We also
Light
call it
dependent.
light dependent. It is also known as with the name of scientist. Hill reaction. Remember, it is also known as the Hill reaction, right? So photo light dependent reaction is also known, or light reaction is also known as the Hill reaction. Clear? And look, so here the light comes and light fall down on the this one, this stroma molecule, right? And this is the granum, right? So granum light falls on. Photolysis of water takes place, means water molecule breakdown, and oxygen is evolved. So photolysis part is called light reaction. It requires the light. In absence of light, it does not take place. That's why the photolysis takes place only in the daytime, right? Now, oxygen is evolved, and along with the oxygen, ATP and NADPH is produced. ATP and NADPH both are the energy currency, means both are the energy. One more thing additionally, which I want to tell you, I will discuss it again and again. I will repeat it again and again. 1 GTP means guanosine triphosphate is equal to 180. Both have the equivalent. Either you call it GTP or you call it ATP. ATP is obviously it's ATP. 1 NADPH is equal to 380. It have energy equivalent to the 380. 1 FADH is known as FADH is equal to 280. Is that clear?
Yes, sir.
Okay. So in this reaction, oxygen, ATP, and NADPH are produced. These ATP and NADPH, they are taken, they are consumed by the dark reaction or C3 cycle. What is the C3 cycle? The fixation of carbon dioxide, means carbon dioxide gas is fixed and it is converted in some solid substance that is called food, right? That is called dark reaction. So in dark reaction, what happens? Carbon dioxide fixation takes place. It do not require light. It's not like that it takes place in dark. It's not like that. Basically, it do not require light. That's why it goes on both day and night. So it takes place in both day and night, right? And it's known as the dark reaction, right? So this dark reaction goes on. Carbon dioxide is converted to G3. G3P is 3 carbon compound. That's why the cycle is also known as the G3 cycle or Calvin cycle, right? Calvin was a scientist who discovered this cycle. That's why it is also known as the Calvin cycle. G3P is first stable compound. That is the glyceraldehyde 3-phosphate. G3P is converted into what? Glucose, right? This is called dark reaction. This ATP NADPH is consumed and it is converted to ADP and NADP plus, right? It again goes back to the this part. So dark reaction takes place in where? And light reaction takes place in where? In gray, right? Tell me, is it clear or not? If you have any doubt, please ask.
Yeah, sir.
Clear? And collectively, both are known as the photosynthesis. Now come to the cyclic and non-cyclic photophosphorylation. Here someone, like in the last class, someone asked me that what is the FAD, what is NADH. So I have written full form over here. So I told you that one FADH is equal to 2 ATP, one NADH is equal to 3 ATP. So FADH is called flavin adenine dinucleotide, right? NADH is nicotinamide adenine dinucleotide. ATP is adenine triphosphate. GTP is guanosine triphosphate, right? So that is the full form. Now come to the cyclic and non-cyclic photophosphorylation. So what is this non-cyclic? Photophosphorylation means formation of ATP. Look, what is non-cyclic? In non-cyclic photophosphorylation, basically electron start its journey from one point and it reaches at another point. Means it do not come back. This is called non-cyclic. In non-cyclic photophosphorylation, both photosystem 1 and photosystem second participate. Both of the photosystem PS1 and PS2 participate. Electron is extruded with the energy of 23 kilocalorie per mole energy. Right. You have to focus only that part where I have underlined, where I have encircled with the red color. You have to remember only those. Try to understand. Non-cyclic photophosphate. So P680 or PS2 that contain oxygen evolving complex OEC. So OEC, it is made up of manganese, calcium, and fluoride. Photolysis of water takes place. So 2H2O breakdown, oxygen is evolved, 4 hydrogen ions are evolved, and 4 electrons are evolved. These electrons are taken by the electron acceptor, right? Electron receiver, electron acceptor. This electron acceptor receive the electron. These electrons jump upside because they have extra energy which is provided by the chlorophyll A molecule and antenna molecule. Now these electron reaches to pheophytin. So what is this pheophytin and all that? They are a type of cytochrome complex made up of protein and some other compound. So electron reaches to pheophytin. When electron reaches from pheophytin, they reaches to plastocunine. From plastocunine, they reaches to cytochrome B6F. I have written over here. Cytochrome B6F. When electron passes through the cytochrome B6F, formation of ATP takes place from ADP. And this is called photophosphorylation. So formation of ATP takes place. Then this electron from cytochrome B6F, it is transferred into plastocyanin. Plastocyanin transferred to chlorophyll P700. Now this graph is coming down. Why? Because this graph represents the energy of electron. First electron got the energy, now the electron is losing its energy. When these 4 electrons will reach to P700, P700 already have captured the light energy. It will provide energy to these 4 electrons. These 4 electrons will again get energy from the P700 and they will jump. There will be received by the electron acceptor. From electron acceptor, they will reach to the FS. IFS is the protein. From FS to FD, ferredoxin protein. From ferredoxin, finally the electrons are accepted by NADP plus. These 4 electrons are accepted by NADP plus and converted into NADPH. So in electron transport chain, non-cyclic, which is the final electron acceptor? Tell me, Aisha and Rabia. NADP plus. If NADP plus is present, it will receive electron. If NADP plus is not present, where the electron will go? No one will receive the electron. Please note it down. In non-cyclic photophosphorylation, in non-cyclic photophosphorylation, NADP plus NADP plus NADP plus will receive the electron, and it is final, and it is final electron acceptor. It is final electron acceptor. Full stop. So is that clear?
Yes, sir.
Yes, sir.
Now, if NADP plus is not present, then what will happen? Then the cyclic photophosphorylation will take place. Suppose that if you want to go your friend's house, you will start traveling from your house and you will reach to your friend's house. But if your friend is not present over there, what will you do? You'll come back. Right? The same thing happened with the electron. When NADP plus is present, it will receive the electron and electron will be consumed. Now, if NADP plus will not present, then electron will come up. That is called cyclic photophosphorylation. In cyclic photophosphorylation, only P700 involved. Look, electron will start from moving, it will get excited, it will reach to primary electron acceptor. From primary electron acceptor, it will reach to ferredoxin, but NADP plus is not present, right? So electron will come to PQ, plastoquinine, right? When electron will come back from ferredoxin to plastoquinine, formation of ATP will take place. Means photophosphorylation will take place. From ferredoxin, the electron will come to cytochrome complex B6F. Again, the energy will produce. Again, the electron will come back to plastocyanin and will again come back to P700 from where it has started the journey. This is the cyclical reaction. That's why called cyclic photophosphorylation. When the cyclic photophosphorylation will take place, it will occur under the condition of low light intensity. When light is low, carbon dioxide fixation is inhibited, means stopped, right? NADPH is not required, right? And halobacteria and purple sulfur bacteria, cyclic photophosphorylation goes. Go through with it and tell me if you have any doubt.
No, sir.
Okay, so everything clear? Now, what is the chemoosmotic? Yes, please.
Sir, can you please say again what is the electron acceptor?
Electron acceptor? Yes, electron acceptor. We'll get it from this one. Photosystem. These are the electron acceptor, primary electron acceptor, which will receive the electron which come from the water. Got it?
Yes, sir. Thank you.
Welcome. What is the chemoosmotic theory? Now I told you when electron will pass through the photocytochrome B6F, ATP will produce. How the ATP will produce? That is the question. So chemoosmotic theory is a theory which tells how does the production of ATP takes place. Try to understand. Please, I want your 100% focus here. Look, this is the structure of what? Thylakoid. This is thylakoid. Thylakoid. So these are the thylakoid, and this is the structure of a thylakoid. So try to understand. This is the thylakoid membrane, right? This is thylakoid lumen, right? When breakage of water molecule will take place, how many electron will be produced? 4 electron. And how many hydrogen ion will be produced? 2 hydrogen ion. Means if there will be 2 H2O, then there will be 4 hydrogen ion. Else from 1 H2O, only 2 hydrogen ion will be produced. These hydrogen ion will remain inside the lumen of. Try to understand. Now, when electron will pass through the photosystem second, it will goes to plastoquinine, and from plastoquinine, when electron will pass through the cytochrome B6F. The moment when electron will pass through, the cytochrome B6F, what will happen? 2 hydrogen ion will be pumped in from stroma to inside the lumen of thylakoid. Again, the hydrogen ion concentration is increasing. Now the electron will go to the plastocyanin. From plastocyanin, electron will pass to the photosystem 1. From photosystem 1, just a minute. Okay. So when electron will pass through the plastocyanin, it will go to photosystem 1. From photosystem 1, pyridoxine, and pyridoxine to NADPH. That is the final acceptor of the electron. Is that clear? Getting my point? Okay.
Yes, sir.
Now the inside the lumen, means inside the space of thylakoid, hydrogen ion concentration will increase. And outside, that is a stroma. In a stroma, hydrogen ion concentration is low. So there is a gradient between the hydrogen ion concentration, gradient of the hydrogen ion concentration between stroma and the lumen of thylakoid. Thylakoid lumen will have more hydrogen ion concentration, and outside stroma will have low hydrogen ion concentration. What will happen? Now hydrogen ion will try to move out of this thylakoid from its higher concentration to lower concentration. Means diffusion of hydrogen, but unfortunately, hydrogen ion cannot cross the membrane of thylakoid. There is only one escape point, one escape gate. That gate is ATP synthase enzyme. When hydrogen ion will leak out from the ATP synthase enzyme, formation of ATP will take place from ADP. At the leakage of two hydrogen ion. Try to understand, at the leakage of two hydrogen ion from this at the leakage of two hydrogen ion, formation of one ATP molecule will be formed. So at the leakage of every two hydrogen ion, one ATP molecule will be produced. This mechanism of production of ATP is called chemoosmotic death. Is it clear or not?
Yes, sir.
Now the question was there. I have seen the question. Question was given, who given the chemoosmotic theory? Can you tell me? Aisha and Rabia. This is a question of the medical exams. They hardly ask you nowadays. They hardly ask you that who is the discovered, what they discovered, right? Can you tell me the name of the person who given this theory? Michel. Michel. So Michel was the scientist who given this. This was the question. Now the NADPH and ATP is produced here is utilized in the Kelvin cycle, right? You have to remember at the expense of every two hydrogen ion, one ATP. Now come to the light reaction. Light reaction is also known as the Hell reaction, right? Water molecule breakdown into two hydrogen ion, half oxygen and two electrons. So basically half oxygen, half molecule do not exist. It exists on the pen and paper, but practically it not exist, you know. So what will happen? Two water molecule will break down, four hydrogen ion will be produced, an oxygen molecule will be produced, and four electron will be produced. This is called photolysis. So photolysis of water takes place in the with the help of oxygen evolving complex at the photosystem second. Oxygen evolving complex is made up of manganese, calcium, and chloride. I have told you that even this question have been asked. This is a very important question, right? So four. So that's how the reaction takes place. 2H2. 2H2. So 4 hydrogen ion and 4 hydroxyl ion. Now these four hydroxyl ion will be broken again under the influence of the this one. So 2H2, one oxygen molecule, and 4 electrons are produced. This reaction is called photolysis of water. Tell me, is it clear or not?
Yes, sir.
This is just diagram of oxygen evolving complex. Production of assimilatory power. So what is the assimilatory power? NADPH and ATP is known as the assimilatory power. NADP plus will take 2 electron and 1 hydrogen ion, and NADP reductase will reduce it, right? Because addition of the hydrogen ion is called reduction, right? So NADP plus will get the 2 electron. So I have already told you who is the final electron acceptor. NADP plus is the final electron acceptor. So NADP plus will take 2 electron and hydrogen ion, and it will become NADPH. Under the influence of the reaction will be carried or carry forward with the help of NADP reductase. Tell me, is that clear?
Yes, sir.
Biosynthetic phase, this is the Calvin cycle. We'll discuss in the next class because today it's not possible. But go through with whatever I have told you today. Go through with your NCRT and let me know. We'll discuss dark reaction. We'll discuss this dark reaction, that is fixation of CO2. We'll discuss in next class. Till the
Yes,
time,
sir.
go through with your NCRT and ask me if you have any doubt in any line, in any question. So are you doing the previous year question paper as well or not? Tell me honestly, both of you, Aisha and Rabia. Like, I'm teaching you, so are you doing the same or not?
No, sir, not really.
Okay, fine. What about you, Aisha?
No, sir.
Okay,
Sir, I
so
actually have exams coming up, so I
you have
started
actually
during the
exams.
holidays. Yes, sir.
When?
Next week, so I'm preparing for that.
Okay, okay, good,
Same
good.
here, sir.
Don't worry. So what I'll do then, why I'm asking, because if you are not doing in the next class onward, like suppose that in the next class we'll complete this part, photosynthesis. So at the completion of the class, I'll give you some sample of question like 10, 15 or 20 question, whatever the question. So in the first page, I'll tell you these are the question, recent year question. So with the help of those questions, you will come to know what kind of the questions are being. That is very important for your preparation. The type of the question, the flavor of the question. If you get the flavor of the question, then definitely you'll do that, right? So all clear, whatever we discussed today.
Yes, sir.
Yes, sir.
So let's meet in the next class, and next class you will tell me that we have to start from the dark section. Fine.
Yes, sir.
Yes,
Okay.
sir.
Bye bye. Take care. Good night.
Thank you, sir.
Yeah. Welcome.
Thank you.